The white laboratory rat is perhaps the most well-known and iconic organism used in biology. The rat in this context functions as a miniature stand-in for a human being. Lab rats are modified by selective breeding or genetic engineering to present diseases and behaviors that more commonly afflict people. This particular rat was bred by Finland's National Public Health Institute to embody one of their nation's most prevalent public health concerns, alcoholism. The rat is a descendant of a population of rats developed at the Wistar Institute in Philadelphia in 1906 that were arguably the first line of inbred animals still used in research today. From this population, the alcoholic rats were selectively bred over many generations by breeding only the rats that preferred alcohol over water when given a choice between the two. In Finland, they were used in the development of the Sinclair method for the treatment of addiction that attempts to chemically reprogram the pleasure response of the brain. During the early 2000s, the research project was canceled, and the status of the alcoholic rat population in Finland remains unknown.
My name is Rich Pell, and I'm here representing this organization called the Center for Post-Natural History, of which I'm, you could say, founder and director and like janitor. But I'm not the only person involved in the center. I would also have to give credit to our lead scientific advisor, Lauren Allen, who's back in Pittsburgh. And here in our audience is Mason Jude, who's responsible for the structure you see upstairs, which is amazing. So thank you, Mason. So I'm just going to go through kind of this basic idea of what I think is post-natural, and maybe that'll help fuel a conversation that we can carry on. Because I think there's some competing, really interesting competing words to describe this whatever it is, this next nature, this super nature I heard. What's the other one? Second nature. And I'm here to talk about the post-nature. So this all started to come about maybe 6 or 7 years ago. I was hanging out in a lot of natural history museums the same time that I was learning about this new kind of emerging form of genetic engineering called synthetic biology at the time. And I just became aware that there were these kind of gaps in the collection of the museum. There are things that weren't there. There are things that I was interested in, living organisms, that were not present in the Natural History Museum. This place that kind of by its own design is there to try to tell a complete story of, like, you know, bring into one place the global diversity of life on planet Earth. But there's this stuff they were leaving out. And it tended to be things that